Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
A large-scale project designed to assess reproducibility in preclinical cancer research has identified significant challenges associated with repeating other scientists' work, renewing calls for increased transparency and data sharing in the biomedical community. The project, launched in 2013 by the nonprofit Center for Open Science, CUS, in collaboration with the online research marketplace Science Exchange, attempted to reproduce key results from more than 50 high-impact studies published between 2010 and 2012. For the remainder of the studies, the team often wasn't able to obtain enough information about the methods used from either the papers or their authors, and had to abandon attempts at replication altogether. The project's conclusions are summarized in two articles published today, December 7, in eLife. A scientific paper rarely tells the whole story, he said. This is a rigorous way of showing that. The project often blamed for challenges in translating animal research to humans. The problem of poor reproducibility has long been a source of concern in the biomedical sciences. The CUS project, which was funded by the Laura and John Arnold Foundation, was one of several discipline-wide efforts launched in the 2010s to dig into the problem and provide possible solutions. Each study would be assigned to a group of researchers who would gather the necessary information to write a registered report, a paper describing exactly how they planned to repeat and analyze experiments. It's like a quality check. Is this the replication rate we would expect to see? And if it isn't, where are we going wrong? Then, the teams would carry out the experiments and compare the findings to those of the original paper. While a handful of these studies successfully reproduced key parts of the original papers, many could reproduce only some parts, or none, or the results couldn't be clearly interpreted. 7 were statistically significant in the opposite direction. An increase in the original study, for example, became a decrease in the replication attempt, and the rest were statistically insignificant, or null, results. Differences in interpretation for researchers who have been following reproducibility issues in scientific research. The results are unlikely to be surprising, says Marcus Munafo, a biological psychologist at the University of Bristol and chair of the steering group for the UK Reproducibility Network. More than providing an assessment of individual studies, these kinds of large-scale projects act as a diagnostic test of how science is working, he says, adding that while the studies were published a decade ago, the project's findings are still pertinent to our understanding of science. It's like a quality check. Is this the replication rate we would expect to see? And if it isn't, where are we going wrong? See, UK Group tackles reproducibility in research. Brian Nosek, COS's executive director and co-founder, offered a similar interpretation at the press conference, saying that the project doesn't provide answers to why a particular result could or couldn't. This last possibility was highlighted by several scientists whose work was selected as part of the reproducibility project. He coordinated with various colleagues and collaborators on the original paper to help the CUS researchers obtain the information and materials they needed, he tells the scientist. But he notes several differences between his lab's 2011 cell study tracking extracellular matrix remodeling in the tumor microenvironment and the COS's replication attempt. For example, in both studies, tumor-bearing mice were sacrificed for ethical reasons once their tumor burden got too large, but this endpoint was met after just 45 days in the replication attempt, compared to 70 days in the original study, a difference that the CUS researchers acknowledge in their See, Potential Causes of Irreproducibility Revealed. Dean Tang, a cancer biologist at Roswell Park Comprehensive Cancer Center whose 2011 Nature Medicine study on cancer stem cells was also selected, is more critical of the project. He tells the scientist in an email that he was extremely disheartened by the CUS team's approach, and contests the validity of the group's replication attempt of his work. In a 2019 letter to eLife, Tang and a co-author write that COS's researchers deviated significantly from our original work, listing several examples of differences in methods and materials. Barriers to reproducibility. Alongside the scientific work, CUS researchers collected data on the practical obstacles they faced during the project. This meant that researchers had to reach out to authors to request information about materials, experimental protocols, analyses, and other details that had been omitted in the published studies for space or other reasons. Discussing possible solutions, McNutt said that better incentives are needed to help boost sharing among scientists. Currently, there's little incentive to cooperate with a replication, she said, particularly when it comes to papers that are already highly cited. Some practices, such as data sharing and code sharing, are already beginning to be mandated by journals or funders, she added. Lauer noted that the NIH will be implementing a new and improved data sharing policy in 2023. However, the speakers also noted that some of the bigger problems are cultural, namely, the scientific community's tendency to view replications as personal attacks rather than as opportunities to advance understanding, and to treat publications as completed products rather than works in progress. The problem is that we focus on outputs rather than process, says Munafo, echoing a point made by several speakers at the conference. That's what I think fundamentally needs to change. Quote, "Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you."